So these are images of what we're going to see. These are cheek cells, onion cells.
저장 안저디 저번들 따뜻 삼바에 빠르 dinner 샤르그레스.
And just thinking about what you've already learned about cells, how can you tell an animal cell versus a plant cell? Just by looking at those pictures.
지신 타본다네 심진 타본이 거라 망고지 장바 텔라 시샤치 결혼서 가기 지신 타본이 가기 심진 타본이 가리 자고 그래. 장제유메 Sopta. Some say the structure, some say absence or presence of chloroplast.
Okay, so certainly the thing that we can actually see in these images is that structurally those cells are quite different.
다이네 갑자기 따뜻한 방 안주디 이입던 꼬바디기들 작으면다.
And you're correct that there are structures in each of these cell types that aren't found in the other, but we can't actually see that with a simple light microscope.
다이네 코니기 바로라이네 다 원타 유메 다 켜버든디 여레스 임바이나인데 미기 무스코 통무투바지 차고레스.
So Phil talked about that this morning, that we have these tools that help us to see things that we can't see with our naked eye, but they have limits.
저자네 여기 자동차 희기네 간저 따투고 말했으라. 저장 가상한저 믹 쟁바기 통무투비야 되는 간저 주체든지 베지탄체네 로바체체 따투고를 라임바. 데 따투비야 될라양 체지레스코. 창마 따투고 말했어.
So how would we describe the difference in structure between plant and animal cells?
저장 코니 딱 꼬바기 켜버되네 간지 가르도 드시겹다스. One is attached to each other. The other one is freely kind of not attached.
Okay, so that's one thing we notice is that in the case of our epithelial cells, our cheek cells, when we get them from the inside of our cheek, they kind of break apart and they're not attached to each other. Whereas in the onion, they're attached.
저선 땅은서 장반에네 디 타본데 렌드 삼바 콘조가선 타본데 디 카토르도 레스 찍지 자르고 말했네. 쫑라 차샤바이나 네 타본 삼마 찍지 자르데이도 왔어.
What else do we notice?
디 마임베 토슈 샴바 가르도. Those different colors.
Okay, so different colors, and that's really mainly due to they use different stains in the two different cells, but they also take up the stains differently.
다네 디 조데 가르스나 코 전제 코라문다 베지탄 레스 다나 신코 전제 집당 주탄대한 코문다 켜버려 레스. 잎. So the shape is different. One is more square, the other one is more round or
Yeah, so that's a distinguishing characteristic or an important difference between plant and animal cells. Animal cells have no cell wall, whereas plant cells have a rigid cell wall that makes them square like that.
저단디 심정이 타본은 지싱이 타본이 바로 있다네 켜버 연하면지 디차고 레스네 심정이 타본라 네 디오 마레스 가서 그래. 저 타본 거란라 디오 마레스 찍바스 남는데 칠로라 다 찍반다 오지르다. 네 심정이 타본라 디오 마르다. 지싱이 타본라 여버체.
So our goal for today is for everyone to create 2 slides. One slide of your cheek cell, one slide of an onion cell.
저단 드린 건서 다네 미유드 가르스나 키란 속에 단다 쫑기네 타봉 다에게 단다 디치서로 실간라드스 디 양안에 서서 담바네네 타봉 렌체드스 다에 니 서레스.
And all of the materials that we need to do this lab are in the back of the room.
저자는 간절로 고에게 잘라 창마에네 거기 감옥이 디 잡로를 샤셔레스.
Be Vanna White. So back here we have the slides, and be careful with them, they're glass. Don't cut yourself.
저단디 쇼고디데 마차로 제스도 또 차에 있는가요?
Your fingers will be blue when you're done.
Yeah, we both have blue fingers. There are cover slips, little clear squares to put over the slide once you make it.
저단 단다 쉬고 간라 양길 쉬고 답답지도 마 this 자리야 춘춘지 그랬어.
There are onions here for us to get the onion cells. To get the cheek cells, you use a toothpick.
저단에 먼저 담바데스 타본 렌드 삼바단다 디 팀베도 팀베 베주탄 거랬어.
So all you do is you take a toothpick and you just scrape inside your cheek and then put it on the slide.
저단다에 팀바디네 스스 카나라 두스네 가서 그래.
You're not trying to draw blood.
두두 단서 그래. 치마 스스 치마 열어 치마 베주탄체. then this 타본 렌드다.
And you do need to put a little bit of water on the slide for both slides.
다는 신타에네 거기 레벨업데 득디 삼바에네 추데스 고레스 추디나 티바 카시지 겹 되는 리얼스.
And then we have the dies here as well.
다 전제되어레스 거기.
I do have an image. Do you want to just pop to the next? These are basic instructions for making the onion slide.
저단디네 거기 쫑기네 실립 단다 소야기 단다 간디지 소고 세기 림바드레스 다 투스투스 치는 소고.
And you can just look at the pictures. The pictures explain what to do.
저단네 파르디들 타네네 코 고마세 버치요 마레스.
Once you've put your cells and stain on the slide, put the cover slip on, and then look under the microscope.
다이네 단다이 지신다운 타본단 쫑이 타본디디 단다이 실 레벨디 간라샤 에네 단다 천제데네 쭉살스나 단다이 쇼고 춘준디 두스카 잡으라 데네 코타 초초레스.
Does anyone have any questions before we get started?
디톨로 티와이베 가리가 강 선데 가리가 강라 시작. Hello. So the question is, should they put the dye before putting cell or after putting cell?
After putting, so put cells first, then the dye, then the cover slip.
담보디네 타본디 단다 빠빠 가리나 타본디 샤로아 디 강라이네 티바게레스 단다 천제디 티바게레스.
And you just need a very small amount of dye.
저단 티바디 테스마도 고마레스 네 천제디 테스마도 고마레스.
Ready?
자리에 왜?
You want to take maybe half of them over there and do the cheek, and I'll do half
Sure.
the onion here.
Okay. So let's divide, and half the room will make their cheek cells first, half will do onions first, and then we'll switch.
저단 담보에는 간서 체가이네 쫑기 빠빠 쫑기 타본데다 쫑기 타본데 따지체 네 체가이 가르스나서 담바기 디 타본데 따지체 인스 전 디젤라 가르스나 양길 this 리미체 인스다.
Yeah. Okay. Right? Or are we going to switch?
I think we'll have to switch back and forth.
Okay.
Once we get the tissue on, they can meet in the middle.
Okay.
Okay, this half, we're going to do an onion. Come on up, I'll show you how to do it.
This half.
다 페로체스 네 체가데 파 체가데 초데건
Slides.
So you don't need the whole quarter, just get a piece of the onion,
They must go more less that's all.
peel back the leaf,
Don't get such a ridiculous chiles.
and then either with your fingernail or with a knife, peel this little membrane.
This.
Can't see it.
King Ling, there are change my go.
Oh, those weren't open. And it helps put a little drop of water on your slide first.
한번 추특바 가시지 겨고레스. 추특바 겹.
I'm not doing You get that off, then you take one of these slides, take the paper out, lay it down. If we, yeah, you want a little bit of water. This is just, that's another dial. Just to get it.
탁. 치마 소스 담박 나로라 this ditch da karsa das yama tutu tanchera.
Then swirl it in the water.
This is the dye.
So like Sunzi there is
One of the cover slips. Put it on.
equal jobs.
So now it's wet. Then go look at it.
And this one,
That's it. That's it.
yeah.
Be very careful. The knife is kind of sharp.
Pesazo não, de trip não é isso. 그다음에 강아지 있지라. 디기서 디기서.
If I can help you, I'm right
디기서
here.
저. 아, 계속 저기 저기는 되게. 지도하고서. 지도하고서. 이동으로 거기 점점점으로 지도. 점점 될래. 제가 루트 리.
We have
I
cotton
wonder if
ball.
that might work. Hey Phil, do you think it would work just to put a little of the dye on?
That might work.
You want to give that a try, and I'll give that a try. Let's see if it works. Yeah, there's a bunch in here.
You mean instead of using dropper?
Yeah.
I was thinking about using it for.
Oh, we could do that too, but we don't have enough, I think, and the toothpicks work. But our slow point is we only have one dropper.
덩그대.
있으면 나왔다.
오.
지치고는 다물어 줄 때.
다물을 먹는 스타. 오호.
Put it on this.
Yes, almost. Yes, perfect.
If you look, you can see the cell walls, but there's no stain, so no nucleus.
These are cells.
Oh yeah. So if you're done looking at the slide you created, you can come make the other type of slide.
따이네 다주 네 르미치네 따라와 네 거기 심정이 타본라 따니도 저 지신기 타본라 따 지신기 타본라 따니도 저 심정이 타본라 따 쇼스
던져. 저는 무지 좋다. 맨날 저 맞아.
일단 다 늦는데 올수 턴 거다.
아직 좀 봐서
근데 dus daarin bij.
다리기. 충분히 남았다. 다리 보여 다리.
Yeah. 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 Great. You see?
근데 다른 거에나 정말에.
Cheek?
Two and maybe this is yeah. One was touch the level. Good for. It's not good.
So this little dot, this is the nucleus.
Yep. That's the nucleus, and in the plants, the nucleus is off center because you can't really see it, but there's this large vacuole right here that pushes the nucleus to the side.
This one.
Yeah, so plant or animals don't have that, so their nucleus is pretty much in the center.
This vacuole, what is it?
It has a variety of roles, but mainly it holds water to help keep it's almost like an internal skeleton, like a water balloon. So the vacuole fills up with water, and that keeps the cell rigid. So you know, like when a plant gets dry, how it droops?
So the nucleus is squished between cell wall and vacuole?
Yep.
Vacuole is this big.
Pretty much, yeah. It's called a large central vacuole. And so you know when a plant gets dry and it wilts, that's because that vacuole is losing water. And then when you water it, water goes back in the vacuole.
어디 말해? 그스까.
Sen 어디 말아?
말해. 네가 담바로.
Attach. Attach. All these cells attached.
Yeah, they're all attached.
These are not attached.
Right, because we scrape them free. Right, when they break apart.
They are never attached each other.
No, in the cheek they are attached. Right, but when we take the toothpick, we scrape them free.
In union is all attached. Yeah?
Right. Yep.
Subuhin allora mi cell pucci na pucci mo 셀 코로나 요로 젠 자고로 셀 코로나 젠 자고로 거의 납서다 셀
유로 도그레스.
So are they aware of like forensics and how when people do crimes they leave cells behind and scientists can use that to figure out who they are? So even though they're normally attached, some do make, so if you spit, some of your cheek cells go with you, or when you touch something, you leave skin cells behind.
저한테 남긴 자르데 그랬어라. 나 따치 치마 유야다 ene lagwa dustukdit sambaro nganzo susu ta ene tabung kashi jipal ko yuk jasha gores 저서 남경 nganzo 집 garso nindo paso dip pejichegido atanda forensics technology migita supugita jelu karakari yomendra jeshil karakari yomedine jama kunzo taji chegido ene tabonde La yona tabonde taji cheche ne Quran kiran ba ne munzi chegido.
And then the scientists look at the DNA and they can say, you were at the crime scene, that was your cell.
지산다 ene 체리batso ide ne DNA ta taji chechero. E kiran didi pagila yobade tada hakku gres.
Is this a cell?
That's the nucleus.
Nucleus. It's a DNA cell.
Nucleus.
What of this?
Nucleus.
So is this the whole cell?
This is the whole cell. That's the nucleus.
That's the nucleus. What of this?
And that's the nucleus of the neighboring cell.
I see. 셀 미더니더 주차사 띠니래서 그 띠띠 김치로 잡고서 담대니 띠띠니 I see.
And in plants, the nucleus is to the side of the cell, whereas in animals, it's in the center.
In the center, yeah.
Yeah, and that's because you can't see it very well, but there's a large water-filled vacuole, and it pushes the nucleus aside. And when a plant wilts, when it loses water, it's out of that vacuole. Then when you put water on the plant, it pops back up because the water goes back into the vacuole.
다 통굿. 따따다.
Situation where they were doing blood genetics, ABO blood groups, and a kid figured out he was adopted.
야, 다 망무드. 다디 나로네라. 디 나로네 ngombujuda. ngombude 타가 타본레. 네 ngom부 마임베 네 타본 셴바드네 망무다. 디 타가마르 타본. So on this slide, the blue one is white blood cell, and the others are red blood cells, right?
That sounds right. I didn't see the blue one. Yeah. Those are white blood cells.
네. 저산 디 나온라인에 셴바드네 망보였어다. 디 따초. So the blue ones are white blood cells, and the others are red blood cells. Right?
What kind of white blood cells? I can't tell.
타거 타봉기는 냉고말라로 냉고말라기 차시처럼 그 나살로 돈렌치고라. 정말 타본데 안저 솔룬킨이 들어. 안저 우탄데서 솔룬킨이데 정말 타본 차고라. 안저 나사 포데가라 나살로 돈렌치긴데 타거 타본 차고라. 말해. 니키 쳐도서야지 타본나로 코란기로 나로라로. 타 먼저 타요. 타기 나올라 타거 타본여래 타모 타본여라. 타모 타본 써야 되는 안저 솔룬킨이들 솔룬. 먼저 우다 안 됐으면 솔룬 키고라. 소보가사가 솔룬 키고. 네 타모 타본 키고래. 나사 포데가라 돈렌체죠. 나살라 돈렌체 냉고말라 냉고말라디. 저한테 타거 타본기네 나살라 돈렌치고.
Got a pinworm. ringworm.
그래 왜래서 저런데 여라 니도 그래 포인트에서
포인트에서 뭐 자가 타고 다 나오는
어디 자가 타버려
그래.
아네 어디 따라지면서 내가 말하지 여리 내가 말하지 자가 타버리라